Hi, everyone. Nice to see lots of familiar faces and some new ones too. I'm Juan Hindo, and I lead World Community Grid. I'll probably be spending the bulk of our time talking about our COVID work, but before I do, I thought I would spend a minute or two introducing World Community Grid to those of you who might be new. So World Community Grid is an initiative out of IBM Corporate Social Responsibility. It runs on the BOINC platform. It's a volunteer computing initiative. And we focus specifically on supporting research in health and sustainability. We actually partner with research organizations. This isn't our science that we're supporting here. So we partner with research organizations at public and nonprofit organizations, and we give them access to the free computing resources that our volunteers donate. Along the way, we also take on the effort of integrating their research applications into the distributed computing platform, and anything to do with recruiting, engaging, supporting the volunteers as well. Essentially, we're letting the scientists do the science, and we try and handle as much of the technology as possible. Right now, we have 6 projects running on World Community Grid. A couple of them have been mentioned in previous presentations, and especially in Marcus's presentation. I'm going to skip over Open Pandemics COVID-19, because that's what I'll be spending the rest of the time talking through. Our Africa Rainfall project is simulating rainfall in sub-Saharan Africa to help scientists produce more accurate forecasts eventually, hopefully. Our microbiome immunity project is looking at understanding the role of gut bacteria in autoimmune diseases like type 1 diabetes. Help Stop TB is a project that's looking at how the tuberculosis bacterium has this kind of outer shell that protects it from the patient's immune system. So scientists want to understand how this works so that they can design drugs to tackle it. Mapping cancer markers is analyzing tissue samples from both healthy people and patients of different types of cancer to try and identify biomarkers. These are sort of genetic clues that can help with early detection as well as with personalized treatment. And then finally, Smash Childhood Cancer Project is looking for treatment options for different types of pediatric cancer. That project right now is paused. It kind of goes through phases, so it's kind of in between right now as the scientists analyze the results from previous phases and kind of figure out what's next. So the nice thing is, as a World Community Grid volunteer, you get to pick and choose as many or as few of these to support as possible. And now I'm going to focus on the first one of these on the list that I skipped earlier, which is the Open Pandemics COVID-19. I want to kind of rewind to this time last year as COVID was happily spreading itself all over the world. We really started to think aggressively about how can World Community Grid play a role here? How can we help? We already had an existing relationship with a research group in California at Scripps Research. Our collaboration with them actually had been going on for over a decade. We've worked with them on HIV/AIDS project and malaria. And specifically, we worked with Dr. Stefano Forli. He's a structural biologist and computational scientist at Scripps. And at the time, he was in the middle of directing his entire lab's resources towards addressing COVID-19, specifically by looking for treatment options for COVID. And so we jumped at the opportunity to help. I will talk a little bit about what goes into actually designing a drug. This will be a little bit of a repetition from what Suman mentioned, so I'm going to go through it and give you back some time, Matt. But kind of similar to what Suman mentioned, the drug discovery process is painstakingly long, 10 to 15 years long, and it starts with a really, really massive search space of millions of chemical compounds, any one of which could be effective as a drug or the basis of a drug. And so the early phases of that drug discovery process are really just about trying to minimize that search space, really make it as narrow as possible, optimize it as much as possible before you continue with the rest of the pipeline of the drug discovery pipeline for things like testing for absorption and toxicity levels before you get into clinical trials and approval by government agencies. So where our Open Pandemics COVID-19 project comes in is that at that very, very early first stages of the drug discovery pipeline, and in terms of trying to narrow down what are millions of possibilities into a handful that are that can proceed through the drug discovery process. And we're using very similar technique to what the BOINC attack team was describing, using specifically molecular docking techniques. So again, similar to what was described earlier, to design a drug, you really need to understand the structure of a protein that's key to the virus, in this case, SARS-CoV-2, and then come up with a chemical compound that can fit or bind to that protein, therefore stopping it from doing its job and hopefully stopping the virus from getting any further. And the analogy we like to use is kind of like a keyhole. So you have a keyhole and you're trying to find a key to unlock that protein target. Now, in the case of Scripps Research themselves, they have developed molecular docking tools that mimic and simulate that interaction between a chemical compound and how it might fit and bind to a protein target. So essentially, that software is simulating and predicting how effective a chemical compound might be as a possible treatment options for COVID-19. And what they're trying to do now is to use those very same techniques to tackle COVID. And so for Dr. Forley and his team, and this is his team here, who for the first time ever had to work from home, they had all of the necessary expertise, everything from medicinal chemistry to software engineering, structural biology. They knew how to do this from a computational perspective. The challenge, though, is that this is very computationally intensive. So there are hundreds of millions of chemical compounds to screen through, and several protein targets. So back to the keyhole analogy, you have a few keyholes, and for each of them, hundreds of millions of keys, and you're trying to figure out which of those keys is going to do the trick. So to do these simulations at this kind of scale, it's really rare that scientists have the kind of computing power that they need, and that's where our volunteer community comes in. In the first 6 months of the project, we had about 70,000 World Community Grid volunteers run 300 million simulations to simulate how 20,000 compounds might be effective against these key protein targets. Of those 20,000 chemical compounds that were screened, 70 were found to be promising, of which 25 are being tested in the lab as we speak. And the base for this Open Pandemics project is just about to get a really nice boost. Just last week, we enabled the project to run on GPU. Of course, this means that the calculations can run much faster than they can on CPU. But importantly, this opens new research avenues for the researchers themselves. So for Scripps, they can, this allows them to start looking at some of the new variants of COVID and expanding the search space in terms of essentially additional keyholes to try and dock chemical compounds against. And also to, in terms of which chemical compounds to screen, this allows them to tackle more complicated, complex compounds as well. The other thing to note here is that Open Pandemics has bigger ambitions that go beyond this particular pandemic. From what scientists have learned from past outbreaks, we expect, unfortunately, pandemics to become more and more common. What's interesting here is that the team at Scripps, the molecular docking tools that they're using on World Community Grid, they're the ones that developed it. Scripps developed Autodoc and Autodoc Vina, which in this case, we're using Autodoc, the simulation tool that's being used for this project. They had made these tools open source a number of years ago. They have become some of the world's most cited and widely used software tools in the scientific community for drug discovery. And what they want to do now is through the experience of trying to apply these tools to COVID-19 and whatever additional tools they're going to be developing for pre and post processing of the data that gets generated on World Community Grid, they want to use all of that insight and knowledge to help develop an open source software infrastructure to help even streamline the drug discovery process even more from a computational perspective. And hopefully this will help scientists address future pandemics much more quickly. The last thing I'll mention is that we also have an open data policy. Traditionally, that open data policy applies to requiring our research partners to make their data publicly available once their work on World Community Grid is done and they've published their findings. In this case, though, the team at Scripps has also committed to making not just the data, but whatever tools they develop along the way, they've committed to making those open source as well, so that hopefully we can see the benefits of the work that's being done through this Open Pandemics project spill wider than the immediate recipients of the computing power. And with that, I will open it up to questions.
Great. Well, many thanks. That's really exciting stuff. And how long? I'll ask the first question. You mentioned some compounds are in lab testing. How long will it be before you have some feedback from that?
We ask almost on a weekly basis. So they're working with collaborators. The lab testing isn't done by the same team at Scripps. The team we partner with are computational scientists. They partner with experimental scientists for the lab testing. There are multiple phases and they want to double check their work. So we don't have an ETA is the short answer.